Welcome to AI for Good, the leading action-oriented global and inclusive United Nations platform on AI. Organized by ITU in partnership with 40 UN sister organizations and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end, as we will hold a 30-minute networking session in the neural network. Here you can meet, ask questions to our distinguished speakers, connect, and chat with the AI for Good community. It is now time to kick off the session and welcome our first speaker. The floor is yours.
Good morning, good afternoon, and good evening, everyone. My name is Matthias Greschel, and I welcome you to today's webinar, co-organized by the ITU WHO Focus Group on AI and Health and the Department of Biomedical Informatics at Harvard Medical School. I'm primarily working for this ITU WHO Focus Group on AI for Health, which is concerned with generating standards and specifications for the use of AI in healthcare. And if you're interested in our work, please reach out or check us out online. We're always looking for people to join the effort. I have the privilege of introducing today's speaker as part of the AI and Health series. And as we were planning the series, his name continuously came up at the top. So we're really proud to have Shalit here today with us. He's going to talk about the pitfalls and potential of using machine learning to personalize patient treatments. By way of introduction, Uri is an assistant professor at the Technion Israel Institute of Technology, where he is doing research in the area of statistics and information systems. He's focusing on how to apply machine learning to the field of healthcare, and he looks specifically at the intersection of machine learning and causal inference and how to bring ideas from causal inference to machine learning. Previously, Uri was a postdoctoral researcher in Professor Sontag's lab, first at NYU and then at MIT. And prior to that, Uri completed his PhD studies at the Center for Neural Computation at the Hebrew University of Jerusalem. Dear audience, please put your questions into the chat, the shorter the better, so I can feed them to Uri. Uri has invited questions throughout the talk. So whenever something's on your mind, put them in the chat and I'll bring them to Uri. And we're looking forward to chat with you in the neural network after the talk. And yeah, now it's time to hand over to Uri. Welcome.
Thank you very much. Thank you for the kind invitation and for the kind introduction. I'm very happy to be here. And as you said, I'm very happy to take questions throughout. I think the audience is very diverse and I expect many interesting discussions. Let me share my presentation. Okay.
That looks good.
Great. Okay, so as you mentioned, my background is in machine learning for healthcare. And when I got into the field of machine learning for healthcare, it was obvious to me that the only use case I had in mind was using patient data to personalize treatment, which seems like one of the ultimate promises of big data in healthcare, and especially important when there are no clear clinical guidelines. Since then, I know there are many other very interesting applications of machine learning in healthcare, but I stuck to my original dream of trying to do that. And the way I imagine this happening is a patient sees a doctor And let's say she diagnosed this patient with some condition, and there are two treatment options for which there are no guidelines saying which is better for this patient. And this is a situation that often happens, and we'll see some examples. And sometimes she really might not be sure what's the best treatment. And in that case, ideally, we'd like some algorithm to kind of pop up, where she can call up an algorithm and say, oh, for this patient with this specific history and so on, I would recommend T equals 0. And the doctor might follow the algorithm's recommendations or might not, but hopefully it will help treat patients better. So this is the dream. And the talk will be about what might it take to make this dream a reality. And specifically, one of the first things we should acknowledge that this type of decision-making requires what is known as causal modeling. We need to know the effect of actions in the world, so we need to understand causes and effects. In this case, the cause would be the treatment, let's say this treatment or another treatment, and again, we'll see examples later, and their consequences, the consequences of taking this action. So we have to acknowledge this causal aspect of the world. And this is especially true if the model we use to kind of train our algorithms to build the data to build our model is what is known as observational data. So data collected from hospitals and clinics and patients themselves, any data that is not collected in a clinical trial. And most health data is not collected in a clinical trial, but it's just collected routinely across the electronic health records. And the challenge with such data, observational data, is that it generally suffers from what is known as confounding. So confounding means that patients that receive one treatment might be systematically different from patients who receive a different treatment, and we need to kind of disentangle these differences before we conclude which treatment is better than the other. And if we model such a system and we build this recommendation engine, really, we can't know if we're correct before we deploy the system. And again, I'll elaborate about this soon. So basically, the idea is this is not an easy problem because we can't always know if we're correct or not. And still, we want to go ahead and try to do this and want to build confidence and avoid harm in these systems. So the vision I have in this talk is that you might be someone who wants to use patient clinical data to build this clinical treatment recommendation model, and we'll propose a best practices framework for doing this responsibly while acknowledging the pitfalls along the way. And we won't be focused on any specific algorithm, but more on like how to use any given algorithm to do this in the safest possible way. And specifically, we'll talk about 3 phases for this. And the first is the identification phase, where we have this structured way of asking ourselves, does the data we have support what I want to do? So often we have this impression, oh, we have so much data, let's go ahead and build these fancy machine learning models, and that's a lot of fun. But sometimes even a lot of data is not good data. And understanding whether the data supports the specific things we want to do is not trivial and is an important first phase before we go ahead. And this is something called, sometimes in the causal literature, the identification phase. If we're satisfied with that, we'll move to the estimation phase, which is where we build our models, our machine learning models, and try to fit them in the best way possible for our goal. And finally, we'll have a validation phase, which is actually intermittent with the estimation phase, where we validate our models and stress test them in various ways, again, to build confidence in what we're doing, because this is healthcare and we really strongly want to avoid harm. Okay, so here is the semi-mathematical formulation of this problem. So we imagine we have a patient called Anna, we have her medical record, and she has high blood sugar, and we're debating between 2 medications. And almost throughout this talk, I will only talk about a binary alternative because that is the simplest non-trivial case. So we're debating between 2 medications for Anna. And the way we think about it is we imagine these 2 futures for Anna. Under treatment 0, we imagine she has some level of blood sugar, which will denote Y0. And under treatment 1, we imagine some possibly different level of blood sugar, Y1, maybe lower. And we need to know both of these futures if we want to make a well-reasoned decision for how to treat Anna. And these are technically, the technical name for these Y0, Y1 are potential outcomes. Goes back to Rubin and Neyman. And the technical quantity we'll be interested in is something known as CATE, conditional average treatment effect, which is the expectation of Y1 minus Y0 conditioned on X. So X is this vector describing the patient. So it's taken from her medical record, her history, anything else, any data we might have about this patient at the decision time. And we want to know which is higher, Y1 or Y0, so we could just look at the difference. We could also look at the ratio. So I don't know how familiar you are with this kind of mathematical notation, but if you're not, what I want you to take away is that K of X is the kind of treatment effect for the patient with features X. So if we know K of X, we know which treatment is better for the patient with represented by the vector. So statistically speaking, K of X is our target, is what we want to estimate. But the problem is we never directly observe it. So we want to observe it to make well-informed treatment decisions, but we never see it because any patient in our data received either one treatment or the other. If they switch treatment after a year, they're not really the same patient, right? They're older by a year, they're after their first treatment. So really, we only observe one of the two, Y1 or Y0 for any given patient. We never observe both. And moreover, to make things even more difficult, the choice which of the two potential outcomes we get to see for each patient is not random because it's determined by how they were actually treated. And doctors generally don't treat patients randomly. They have the reasons, maybe misguided reasons, but they have usually some reason for why they give one treatment versus the other. So the half of the case that we reveal is not random, but has many reasons, some of which are not seen, we do not know. Some are opaque to us because they go on in the doctor's mind. Any questions so far?
Uri, I can't see any questions yet.
Okay, great. So this is just the introduction. So again, the cake is what we're interested in, conditional average treatment effect. And just to kind of drive this home a bit more, the one way to think about this quantity cake is what if we forced patients with treatment X to receive treatment 1 versus what if we forced them to receive treatment 0. Forced as in irrespective of how treatment is actually given in the world, we say you have to think T equals 1 versus you have to think T equals 0. If we know the difference between these two, then we're good and we actually know how to optimally treat these patients. But as I said, it's not easy to actually estimate this quantity because we don't fully see it. We see only kind of this partial biased view of this quantity. So essentially, we can never provably know what if. We never know for a given patient what would have happened had we treated them differently. Okay? So that's a really important point. So we will never know for a given patient in our data, if we treated them differently, what would have happened. So for any recommendation algorithm, any treatment assignment algorithm, we can never provably know what would have happened, unless we actually go and deploy it and actually treat patients according to it, which of course is a high bar. How do we translate this case to recommendations? So just to make it clear, we will estimate K, so this is K hat, our estimate for K, and present recommendation to the doctor. For example, if blood sugar, we want lower blood sugar is usually better for most patients. So we will say if K hat is negative for this patient, we will recommend T equals 1. If it's positive, we'll recommend T equals 0. Or maybe we want an option also to defer, to say, okay, our algorithm isn't sure, there's high uncertainty about K, so maybe regrettably our algorithm in some cases won't be able to help. This is another aspect of safety. Not giving recommendations, we're not sure. Okay, so how do we estimate this quantity? So I said it's difficult, you only see part of it and so on. There's a ton of algorithms. There's been a deluge of papers showing many different machine learning approaches to do it. You can also do it with like more traditional methods, such as even linear regression sometimes. The problem with all these algorithms is that they only work under some strict set of causal identification conditions. And here we go back to that phase 1, which I mentioned, identification, which says, can the data even do what I want? So every one of these articles, these papers here and many more, start with, we assume A, B, C, D, E. But these assumptions are really non-trivial. And the most important assumptions are that there are no hidden confounders and that there's common support between different treatments. And I'll explain these. So no hidden confounders mean that all the factors that went into the decision that might affect the outcome are actually measured in our data. And that is not always the case. So for example, in psychiatric treatment, a lot of the interaction between the physician and the patient, which might affect the treatment decision, might not be recorded for privacy reasons at all. So we might not have this important data relating to the choice of treatment and possibly the outcome. And in that scenario, we might have hidden confounding. So all these algorithms assume that there's no hidden confounding, that we've measured all the potential causes of the treatment and the outcome. Another assumption, which is slightly easier to verify, is what's known as common support. So we're only dealing with patients who can plausibly receive, let's say, each of the two treatments under consideration. There's a third one, which I'll skip. So these assumptions are not entirely testable. Specifically, the no hidden confounders assumptions, there's no statistical test which will tell us, oh, we've measured all the confounders. So we can't just get a data set, run a test and say, okay, this is satisfied or not. We have to reason about it, but there will never be kind of a strict final answer that says, okay, we're good. We can only be as sure as we can, like we could try to be as sure as we can, but there's no kind of guarantee. And just for people who are familiar with supervised learning, even supervised learning, and a lot of these algorithms are kind of versions of supervised learning, will not work unless these three conditions. Okay, so what's the difficulty? All causal effect estimation methods, which we need for treatment recommendation, rely on these causal identification assumptions, some of which are provably untestable from data, prominently the no hidden confounding assumption. Okay. So I mentioned one hidden confounding scenario in psychiatry. There are more. If treatment is assigned based on some unrecorded patient preference. If there is data which was available to clinicians, for example, some imaging data, which we simply do not have for various technical or regulatory reasons. All of these scenarios are scenarios where we have hidden confounding and we have a problem. Okay, so that's the challenge. The second challenge, which is related, is that we have no test set. So in machine learning, we usually have a test set where we can test our algorithm and see, okay, how well does it perform? And then we have this kind of guarantee. We say, okay, 85% accuracy. If we're happy, we're happy. If we're not, we at least know we have a problem. Here, we don't have a test set because whenever we make a recommendation, which differs from what happened in practice, we can't know what would have happened. We can't provably know. We can hope to model it, but we can't provably know. So these two challenges stand in the way of anyone who wants to use machine learning or statistical tools to personalize treatment, especially from observational data. Okay? And of course, this is a high stakes scenario. Even some pilot system might cause harm, and so we really need to be careful. So my research agenda in this field is how can we still build confidence in deployed treatment recommendation systems? So I told you how this is difficult and complicated and scary, but still we want to move ahead and try to help the doctors. We want to try to safely help them. And I think there is some hope. I think not all is lost. And towards that goal, we developed this framework, which is basically a bunch of a series of steps that we recommend for practitioners to follow to help them build confidence when building such a system. So each of these boxes has kind of a full explanation. I will not go over all of them. I will go over some which I think might be of more interest. Now, before I move on, just want to ask, are there any questions so far?
No worry. I think everyone's used to keep all questions.
Okay, fine. If there is something, so feel free to interrupt.
Okay.
Okay. So I'll go over some of these boxes here, and they're colored according to the phase. So we start with identification, and then we have these alternating estimation and validation phases. So the first thing is really understanding, as I said, can the data even support what we want to do for the specific case? So we have a specific scenario we have in mind, and we call that the target system. So the target system is where we define exactly the setting and context of the treatment recommendation system. So we talk with the clinicians and understand where in the clinical workflow will they need actually the support. So might seem obvious, but when you go down and do it, it turns out to often be highly non-obvious. There's not always a specific point where this happens. It might differ between different doctors or systems or wards or clinics. And defining this really helps, including understanding what data will be available to the algorithm at the moment it is asked to make a recommendation. So some points that help this discussion with clinical partners are, for example, is the treatment decision made by physicians at a well-defined point in time? Is the set of possible actions small? If the set of possible actions is large, if they're debating 100 different treatment options, that might pose a statistical challenge. It might be very hard to estimate one of 100 possible. Are there clear clinical guidelines for the decision? If there are clear clinical guidelines, then our system will have a problem going outside of them, because if all the patients are always treated systematically the same, we don't have any data to support what if reasoning. We can't ask what if this patient were treated differently, because this patient and all patients like this patient are always treated the same if the guidelines are actually followed. So we need to understand this. Is there high variability? So there might not be any official guidelines, but maybe essentially in this hospital, these patients are always treated the same way. And again, we can, using data from this given hospital, we can never ask counterfactuals, what if questions, because all these patients are treated in the same way. So we need this high variability. Moreover, that if there is high variability in treatment decisions between physicians, it's kind of a clue to us that they might need help, because maybe they're really not sure what they're doing, and then an algorithm might help them and make better decisions. And finally, do the clinicians agree? What do we want to optimize? Like, what are the good outcomes? Because that is also not always the case. Maybe the outcomes are the desired outcomes are highly patient-specific and require kind of a lengthy discussion. And then it will be harder to build a system that like ours, which optimizes for one or two, or a small set of outcomes. So all these points really help clarify this target system. And from my experience, often these iterations are really important. They change the system we end up building. We often end up not doing what we thought we will be doing when we start these discussions. Once we're satisfied that we can define a target system, that there is a well-defined point in the treatment, in the clinical workflow, where help will actually be needed and can be supported, we want to understand, does the data actually help? So we need to understand the confounders. We need to see whether they're measured mostly, whether there's temporal separation in the data and what data is recorded before or after treatment assignment. Because if some data don't have a good timestamp, so we might not know if something is a reason, is a cause for the treatment or the consequence of the treatment, and we want to be able to differentiate them. In this phase, something called the causal graph might help. These are well-known, often associated with that Pearl's work. And if you're not familiar, I'm sorry, but I won't be able to fully explain, but these basically map out the relationships between the measurements, the treatments, and the outcomes. And they're a very good cognitive aid to see whether we've measured the important factors, the important confounders. These graphs allow us to kind of formally identify confounders. This is an example of a graph we built with our clinical partners at the Elite Health System for the task of second line diabetes treatment for adults. So these causal graphs also we find help in this identification task. This is a point where we might stop. We might say, okay, sorry, there's no identification. We found using this graph or with the discussions that there's an important hidden confounder which is not measured. And then we need to go back to the drawing board. We need to either obtain better data, change the task, or sadly, sometimes we just have to tell our partners, we're sorry, but we can't help. That we need to acknowledge that is sometimes the case. If we're satisfied, we can go to the next phase, which is estimation. So here we actually try to estimate the case using one of the many models that we've outlined, like I kind of put on the slide earlier. We also estimate something called the propensity score, which I won't go into. And I won't go into the details here. These are the algorithms. It's a lot of fun developing these algorithms. And there are many great methods developed in the last few years. And I'm happy to talk about those offline. Once we have these models, the Kate and the propensity score. So I will mention the propensity score is modeling just the treatment assignment process. So Kate is which outcome will happen under each possible treatment, and that allows us to choose the best treatment. Propensity score is this aid which just models the treatment decision process. So what is the probability that a certain treatment will be assigned to a certain patient in the data? So this describes what clinicians actually do in the data. And as we said, we have no traditional test set. We can't evaluate test error, but we still, often these Kate models have under the hood some regression and classification model, and we can still evaluate these using standard metrics. And if we find that our model, if we treat it as a regression model, performs very, very poorly, then that's a warning sign. It means we might have a lot of variance which is not accounted for, and we might need to be careful. We can compare different Kate models to see whether they're consistent between themselves. If we try two different Kate models and they give completely different recommendations, that is cause for concern. That means there's some, again, variance or confounding here which we haven't accounted for. We could try and apply interpretability methods to make sure that these models rely on things that make sense and not on anything which is too spurious or weird. And we can check these models with the clinical experts once we apply the interpretability methods to see if they make sense to them.
Uri,
yeah.
Can I just briefly ask one question? Can you maybe clarify how does your propensity score and the Kate differ from fitting a linear model? Because as I understand what you've presented so far, so you have an outcome, a binary outcome, and you fit a linear model to it, and one of your variables would be treatment A or B. And so that coefficient essentially will tell you the effect of the treatment on your outcome. How does that compare to what you measure with the propensity score, for example?
So what you mentioned is one method to estimate Kate, and actually it's a method that is, it has some drawbacks. So there are methods, here's a different thing you could do. You could fit two different, let's say, XGBoost models, so boosted decision trees, for Y1 and for Y0, and look at their difference. Or you can apply a Gaussian process to the interaction of X and T and look at that. So it doesn't have to be a linear model. And I had that slide with just a bunch of names. There are many, many different algorithms for Kate, and linear is usually kind of the simplest one. So you start with a linear model, but usually you do want a nonlinear model for these things. For propensity score, it's the same. So it's a binary classification thing. You can use logistic regression, you can use XGBoost, you can use a neural net, depending on your data. The problem is that it's hard to choose, because I could try a linear model and can try the XGBoost model, and I can look at their error, but that's not their Kate error. That's just their kind of ordinary error. It's not the error in saying what would have happened had I been treated differently.
Okay.
So it's still, if any model has like a terrible MSE or AUC, it's a bad sign. It says it doesn't it didn't capture anything of value. But if two methods have similar MSE, it's not obvious that the one which has slightly lower MSE is actually making better recommendations, and there's no way to know for sure, which is kind of vexing for someone used to machine learning, where you can at least choose your model based on the test set, and you can't do that here.
Okay, thank you.
Sure. It's a very important question. Okay, so I mentioned these. So basically, we don't have a good way to test these, but we have all kinds of proxies that we try to look at to see if these models make sense. And again, at least kind of throw away models that are obviously bad. And again, we might have to start again. Like maybe our features just have no information actually about the outcome, and it's completely unpredictable. That never happened to me, but can imagine it happening, and then we might need to start again. How much more time do I have, just so I could?
You have. It depends. So 30 more minutes. But if you leave 10 minutes for discussion, then they'll have 20 minutes.
Okay. Another thing we want to estimate is the deferral. So that is the set of patients for which we have no recommendation. because of maybe estimation error. We have maybe there's a rare type of patient for which we're really not sure about our model. Maybe there are patients where we're more worried about hidden confounding. So I said if there's hidden confounding, we're in trouble. But hidden confounding is not like a binary yes or no thing. It is in degrees. And you might say for this subset of patients, we are more worried about hidden confounding. They maybe at that year recording of data was a bit worse. Maybe they have this special attribute, which means something else happened, which we're not sure about. So we want to kind of estimate our uncertainty for all of these reasons of estimation error, noise, and causal error. And if our uncertainty is too high, we might want to defer the decision. And now modeling all of these is challenging. We have some work on that, but I won't go too much into it, but work that tries to model all of these sources of error jointly for this case. And again, if the kind of joint interval of uncertainty crosses zero, if we're not sure if Kate is positive or negative for this specific patient, we might defer the decision. Finally, the last element of estimation, which I want to talk about, is what's known as policy value. So this is the ideal metric we'd want to use if we could, if we can. So, and I'll explain. This is not a metric of error, it says something else. We're actually recommending actions in the world. So this is a measure of how good the actions we recommend. And it asks the following: if physicians exactly followed our recommendations, so they just did exactly as our algorithm says, what would be the expected outcome of the patient? So for example, if we're thinking about blood sugar and second-line diabetes medication, you might say, the average blood sugar of patients, if they were treated by our algorithm, would be this much lower than the average blood sugar of patients right now if they're treated by physicians. So this is what is known as the policy value. And this is also used in reinforcement learning and so on. So the policy value of the current policy, which we call the doctor's policy, is just the average outcome in the population. This is what actually is happening in the real world. And ideally, a good recommendation would have a better policy value than the doctor's. Now, estimating policy values is itself a challenging causal problem because, again, it relies on this unknowable, where wherever our policy differs from what happens in practice, we can't know for sure what would happen. We only have our model, and our model is only as good as its assumptions, its goodness of fit, and all these issues. So it doesn't get around the problem of confounding, but it is a really important metric that we're trying to estimate, and we'll see examples. And ideally, we want as high a policy, or low, I mean, it depends. Sometimes you want high, sometimes low. We want to optimize the policy. So now I'll present some preliminary results, and this is work led by graduate students from Gutmann, from Technion, and Shimon Sheva, who was at Technion, now at Causality AI. And the first test case is joint work with the Research Institute, where we investigated the effects of two second-line diabetes medications on type 2 diabetes patients who have not responded well to first-line therapy. So first-line therapy here is metformin. And the goal is to reduce blood sugar measured in HbA1c. We had more than 50,000 patients, more than 200 covariates, the demographics, lab tests, and so on. Shalit has a very good data system at their disposal. So we ran our pipeline. I won't go over all of it. We drew that causal graph I showed earlier. We were satisfied that we've measured all of the important confounders. We talked with clinicians at Shalit, trying to understand how do they prescribe these medications, what drives their decisions, seeing that it is actually reflected in the data. And what we found is, for a Kate model based on XGBoost, is the following histogram of Kate values. So you see that they're a bit shifted to the left. So one medication was slightly better than the other, but most of these Kate values are not clinically significant. So if one medication has, it should have like 0.1 HbA1c better than the other, it doesn't really matter. There is a small set for which really one treatment was kind of profoundly better than the other. So specifically, we ended up giving recommendations for 25,000 patients. We had kind of patients drop out for various reasons along the way, so I'm not going into all of this. For about 9,000, we thought that sulfa was the best medication. For 16,000, DPP-4. And we looked at disagreement with actual practice. So we think that 8,500 of these 25,000 wrongly received sulfa, and this is in quotation marks because, of course, we don't really know what would have happened. Only about 1,000 we think wrongly received DPP-4. And what we found, which gave us a bit more confidence, is that we found that most of the wrong sulfa were treated by general practitioners as opposed to specialists. So it seems that specialists tend to agree with us more and tend to prescribe DPP-4, which was a slightly newer medication, not like, but a newer medication. So another way to parse this is look at patients who actually receive sulfa. And our algorithm, our model thinks that about half would benefit from DPP-4 and only a very small percent should have stayed on sulfa. And for many, we have no recommendations. That's the deferral. On the other hand, for patients who actually receive DPP-4, we also think that most of them should receive DPP-4 and only a small fraction should have received sulfa. So what we end up finding in this case is that it's not obvious that there's a reason to even personalize, because the policy value, which I haven't shown here, for basically just moving everyone to DPP-4, I remind you, so for most patients, we just recommend them to be on DPP-4. That's probably the best policy. Okay, so just moving everyone to DPP-4, don't need for fancy algorithm. So I do like fancy algorithms, but reality is what it is. And this is, of course, clinically much better, right? Because it's much easier to just say, fine, just move everyone to DPP-4, stop treating patients with sulfa. That is so much easier to implement, of course. Now, since then, there are new medications introduced, and we still need to gather data and follow-up data to actually kind of evaluate them. So we tried, but we didn't have enough data to support making any meaningful inference about this. So this is one example of our pipeline, which we ran kind of in the chronic medicine case. We also have a preliminary result in the acute disease case, where we were looking with our partners at Rambam Medical Center, that's the Technion University Hospital, on the causal effect of diuretics on kidney function in hospitalized acute heart failure patients. Okay, so this was Dr. Orne Kaspi and Professor Doron Aronson from Rambam and Technion. So let's parse this a bit. These are patients in the hospital. And these are pretty ill patients. About a third of them would die within a year. Is there a question for everyone?
Yeah, one question to the diabetes example from Julian.
Yeah.
He's asking, how do you model time and how do you think about time when thinking about causal inference? And his example is the glycosylated hemoglobin. You likely had many measurements for each patient. Do you just take the first, yeah. The way that
that's a very good question, which we spend many months dealing with. Yes. So eventually, what we ended up doing is looking at 6 to 18 months. And we tried various things. We tried averaging over this time. We tried the last measurement. We tried modeling the entire time course. The problem is that most patients have, let's say, I think at most 2 or maybe 3 measurements. It's not like they're not measured continuously. So we ended up settling for some aggregation scheme over a time window, which our clinical partners said was reasonably okay. Ideally, what we would do is model the entire time course. So model everything, let's say, from 3 months to 18 months, and then decide based on that. But it's harder to make decisions based on this kind of complex object versus just a single number. So we reduced it to a single number. But let me say, that's not kind of the last word. You could do better than just have a single number and try to reason about these more complicated objects. It's a trade-off, because the single number, of course, it reduces some of the complexity, which is both good and bad. Basically, you lose some of the resolution of the treatment decision process, but you might be able to reason about it more easily. So yeah, it's a good question, and we spent a long time trying to figure it out.
Thanks, Uri.
Okay, so for the acute heart failure case, so as I said, these are patients in the hospital. About a third of them die within a year of the hospitalization. So these are really sick patients. And some of these patients develop kidney injury during their hospitalization. And one of the possible treatments for that is diuretics and blood pressure medications. And we had about 2,000 of these patients which had kidney injury. And again, we have many, many covariates and positional confounders from their hospital record. And empirically, we focused on the diuretics aspect, and about half had increased. That was what was prescribed to them. So for about half, they had their diuretics increased, and about half decreased. And our clinical partners at Rambam said they have very poor guidance about how to actually do this. So our P=1 would be decreased diuretics, and this is often improved kidney function. Basically, there's less, they need to work not as hard. So probably that might hurt cardiac function. So doctors actually need to do this kind of balancing act of multiple outcomes, which makes this difficult. And for now, just as a preliminary work, we only examine the effect on kidney function. And ideally, we would like to present the doctors like these two things. Say, okay, if you do this, this is what will happen to kidney function. This is what will happen to cardiac function, and try to make this trade-off and help the clinicians make this trade-off. The problem is that cardiac function is it was more difficult to define and get the data, so we started with kidney function, which is more easily defined in terms of the data. So the question is increase or decrease diuretics, and now I'll present the policy value. And policy value, the units here are in percent return to baseline in creatinine. So creatinine is a lab value that measures kidney function. In this case, 100% would be full return to baseline. So they've recovered from their kidney injury. 0% means no improvement, they're still in a bad state. So here, higher is better. So we ran our pipeline. Again, we did all these things for many months. And as usual, there are all kinds of complicated questions which are not exactly covered by what I talked, for example, this timeline thing. And we took a held out set of 530 patients and had recommendations for 481 of them. So for 59, 59, sorry, we were like, we don't know. Sorry, it was 49. And we estimated policy values via something called the doubly robust estimator and bootstrap confidence intervals. And these are our results. So if this is what we believe would have happened, if we increase diuretics to everyone, their situation would be quite poor in terms of return to baseline of creatinine values, less than 20%. This is what happens in practice. Okay, so this we know is true. So it's a bit more than 20% is what actually happens now in terms of the way these patients are. This is if we randomly assign increased or decreased diuretics. And as you see, it's more or less the same as the doctors. And we were a bit worried when we saw this, but the doctors really reassured us that this could be true. They're not sure that they're doing better than random in terms of the kidney function. This is if we decrease diuretics, everyone. And this is obviously better, but as we said, we don't really want to decrease diuretics to everyone because that might hurt their cardiac function. And these are two of our policies. We have like a more conservative policy and a less conservative policy. The more conservative makes recommendations for less patients. And as we see, their value is higher. So let's parse this. They're better than current practice. So if we compare our kind of machine learning policy to current practice, it's definitely better, p-value of 0.002. It is not statistically significantly better than decreasing to everyone. However, our recommendations only decrease for 43 or 45% of the patients. So we allow more flexibility with respect to the other outcomes because we don't really want to decrease diuretics to everyone. So the next step would be actually to model this effect on other outcomes, which is, as I said, it's a bit more, needs a bit more work in terms of getting the data. It's a bit more difficult. But this is encouraging in terms of promising better outcomes with more flexibility to the clinicians using this kind of personalized policy. Now, I have this graph, but should we believe it? And again, kind of going back to my original point, we don't really know what would have happened if our recommendations would have been followed. In any case, at least when our recommendations differ from what actually happened, which sometimes they do. So we can't know for sure. And the only way to truly know if our recommendations are useful is to run an experiment testing the system with real physicians and real patients having this recommendation system. And that is our kind of ultimate goal. But of course, there are regulatory kind of hurdles we need to overcome. And the goal of this framework, which I kind of briefly presented, is to come into this trial in the best possible and safe shape, right? So kind of being as sure as possible that we're not doing any harm, that we haven't missed anything important, and that we have a system which has a good chance to actually help clinicians. So I just want to thank, again, Rome and Shimon, the students who did most of the analysis, Oren and Doron from Rambam Hospital and Technion, and my colleagues at the Lead Research Institute. And I want to thank everyone here on this video for their attention and time. And I'm happy to take any questions.
All right, thanks a lot, Uri. That was really fantastic. I invite all of the listeners, please shoot in with your questions, and I'll kick off discussion. There's already 2 coming in, but I'll ask this question, which has been on my lips all throughout the entire talk, which is, so how do you, so when you have the models and you want to have a trial in a hospital, what's your plan of integrating the models with the hospital data system? Like, your model needs more than 200 variables, and are all of those available at 3 a.m. in the morning when the physician would like to have his prediction on whether to stop diuretics or continue them?
Yeah, that's of course one of the major challenges. Luckily, we actually have this a new Center for AI in Healthcare, joint for Technion and Rambam, whose entire mission is to help us get the data out and then get the models in for Technion researchers in general. So, like, one of the premises is that a way to integrate models with the IT system of Rambam. Luckily, so the system is not new, but it is in-house, the IT system for the hospital. So that gives us a way to kind of merge on our own terms. We don't need kind of outside help. So it will definitely be a challenge, but this new center gives me hope that we will be up to this challenge. And of course, I'm not the only researcher trying to integrate systems into Rambam. There are several others doing in various departments, various specialties. So we will learn how to do this together, and let's see in a few years if we've actually succeeded.
Okay, yeah, I think you're in the best of all possible places. And I think, yeah, I think the entire world is thankful to Israel and the UK, where, like, I guess most of the evidence has been built on how to navigate through COVID because of the good data infrastructure. All right, I'll give floor to Shirin Elizabeth Korsandi, who's asking, could the use of a digital twin be of use in testing treatment outcomes?
So one of the things, these CAPE models, many of them, what they implicitly, some implicitly and some explicitly do is build this digital twin. And there are other methods to do this. I haven't mentioned, but there's a method for CAPE estimation, which is called matching, which is find a similar patient which received the other treatment. This is often not as good in high-dimensional data, but it is, sometimes it's good. All of these methods do not overcome the fundamental problem, which is maybe a patient seems similar to me and received the alternate treatment, but they're not really similar. They're different in ways which are not recorded in the data, in systematic ways, not recorded in the data, and then I might be wrong. So a digital twin, for me, is a subset of the CAPE estimation methods, which is helpful and useful, but doesn't overcome the fundamental problem which I've described.
Okay. Venet Osmani is asking, learning the most optimal treatment policy can be done with reinforcement learning. Is this something you've compared your work to?
So reinforcement learning and causal inference have close relationships. I also have work in the field. reinforcement learning using observational data suffers from all of these problems. So it's not, there's no way around it. And reinforcement learning tackles, in general, a more difficult problem, which is you treat, you see the outcome, you change the treatment, and you go on and on. Here we're talking on an even simpler problem, just treat at one point, like there's no feedback loop. So we can't directly compare the reinforcement to most reinforcement learning because they're dealing in a more difficult situation, but they're not safe from these problems of confounding and not really knowing what would have happened. I recommend there's a great paper in Nature Medicine from Gottesman et al. About guidelines for safe reinforcement learning in healthcare, which goes into these issues of how to deal with what I mentioned, like hidden confounding and all that in reinforcement learning. Because people who come from reinforcement learning, for example, who are used to working on Atari or Go or Mujoku, aren't used to thinking about these problems which are off policy with hidden confounding. And that paper from Gottesman et al. Kind of lays it out. So that is one part of my answer. The other part is that what we're doing is kind of easy reinforcement learning in terms of there's no feedback. So we're doing something which is, in that respect, easier than classic reinforcement learning and not directly comparable because we have no evolving state.
Okay. Uri, do you mind stopping screen sharing?
Yes.
Your video is lagging a little bit on my end, just to save some bandwidth. Okay.
Yeah.
Then, so when you reach out to your clinical collaborators, how much fluency do they have in terms of thinking about data and the use of data to infer new knowledge for your models?
It varies. The partners I work with are all very comfortable with data and models. They might not have any formal training, but they have some actually do have formal training in machine learning and statistics, some don't, but they're all experienced in working in doing research and quantitative research. If there are machine learning people in the crowd, my suggestion is find a partner you're comfortable with. That is the key to success. Like a good clinical partnerships are crucial. Without them, there's no way forward. So you need the trust and a common language. That has been crucial for what we've been doing.
Okay. Julian has another question. He's asking, when considering existing clinical guidelines, aren't those also based on statistics and may have suffered from the same pitfalls? Yes.
So here's a situation that sometimes that has happened to me. A clinician approaches me and say, "This is the guideline. I think it's wrong. I think I think we could do better." And that very well might be the case. But if in the data I have access to, everyone is treated according to the guidelines, then the methods I described here can't help. Because I have no patient who received some alternate treatment to show, oh, they did better. So other methods might help: biological understanding, in vitro, animal models, all kinds of things are great science, but the tools I described here can't help unless there are people who don't follow the guidelines, which also sometimes happen. The guidelines aren't always as strict as described, or patient or physicians don't always adhere to them, and then we might have some way to get some progress. So it depends on that.
Okay. All right. So Bennett has been asking if you could link to the Nature paper you mentioned, so we can do that later on in the neural network when we join all of you guys for some chatting. And I think it's also a good moment to maybe close off with a final question, and then we can join everyone in the chat to discuss there. And so I'd like to close with the question. So the field of sort of statistics, machine learning, and so forth has really seen a lot of advances. As you mentioned, all of these models are now possible. They're coded in nice packages for many people to use. But then once you apply the models to real world data, you often see that it's really challenging to inform clinical practice. You may not see an effect. You can't even explain what you see. And finally, the real proof will be a clinical trial, which takes years and is super expensive. So what's your general outlook on ML in healthcare now that the time has come for it to be proven, in real world settings and clinical trials? Do you expect the revolution or all these positive news over the past 5 to 10 years will continue, or do you think it'll be less so?
Good question. We're definitely at the place where everyone knows we have to start doing trials, not just for what I described, for diagnostic systems, for all of this. There are not as many as I'd like. Eric Topol, right, had this review about a year ago showing that. I think I'm afraid that the meeting reality will be tough, and that there will be, and we're already seeing it a bit, right? Some of the our expectations or hopes will be dashed, but we have to. I don't know that. That's my guess, but we will have to learn as a field to work with that, right? So pharma has learned it, right? They know to run trials, to be disappointed, to do it again, to learn from it. I'm not sure machine learning in healthcare has learned it yet. We haven't had the option, the chance. And we'll have to learn how to do this and learn our lessons. There's no way around it, right? That will be our tough meeting with reality and learning what's the next step. I think we don't really know how good, for example, clinical records are for doing these things. So not yet. And for example, maybe the lesson will be we'll have to record better. We have to get more data. That seems plausible to me. But I'm still optimistic. I mean, I think it won't be easy, but we will learn a lot and eventually see what works and what doesn't, and learn how to help people. And there are, what a criticism I have of my own work is that I try to focus on something which is really high stakes, and that means it's really hard to do. But there are lower stake things, right? You can help the workflow, make things more efficient, make things easier for the clinician. So many of my colleagues are doing things which are much easier to actually implement and help people now, not after a trial of a few years. And those things have successes already. So that is a good first step.
Okay, thank you. Yeah, that was a really balanced outlook. You mentioned Eric Topol, and that brings me to the announcements of our next webinars in the series. So we will have, in about 2 weeks, we'll have Alex Hanna, who they worked at Google and now joined the DARE Institute, founded by Timnit Gebru. And that promises to be a very interesting talk about hidden bias in data that we also briefly touched upon, hidden confounders that you just don't know exist because you don't measure them. And end of the month, end of April, we'll have Eric Topol speak here. So that'll also be potentially very interesting. So I ask all of you, please share it with your friends and networks. And so I'd like to thank Uri Shalit again for this fascinating talk. We'll be online in about 2 minutes at the neural network, so we can chat back and forth. There will be a little poll for everyone that pops up where you can say if you recommend this talk and the session further. And thank you again, Uri. Thanks to AI for Good, to the co-convener Switzerland, and looking forward to network in the neural network in a few minutes.
Bye, everyone. Thank you very much. Thank you.
Thank you for participating in today's AI for Good session. We hope you've learned something new, innovative, and engaging in today's event. We now encourage you to continue the conversation on the live video wall in the neural network. Here you can ask questions, like and comment, share links, complete the satisfaction survey, connect with interesting profiles, or speak one-on-one using the chat and video function. After the 30-minute networking session, we invite you to explore the lobby, try the smart matching quiz, visit the virtual exhibits and poster boards, and build your personalized AI for Good program. It was a pleasure learning with you today. See you at the next AI for Good.